Okay. Good morning. We from the group of four will present an idea engineering assignment in which the precision assignment is a project text. Previously, my name was Scaltus as a group of four. Here I am with my colleagues, namely Cahya, Revi, Hadi, and Yanti. To shorten the time, we will strike right away. The first one will be researched by Hadi.
Chapter 1 Preliminary Background. Microbe is microscopic living organism which is closely linked in human life. One of pathogenic microorganisms such as Pseudomonas aeruginosa may cause diseases. Sulfur of several plants are bioactive as antimicrobial. The properties of sulfur allium CPL leads to assumption that it has antimicrobial properties. They are Therefore, this study involved the antimicrobial test. The determination step of antimicrobial alum CPL extract consisted of the alum CPL extract preparation, microbial regeneration, and antimicrobial test. The, the antibacterial determination we are performed by by paper Dix method based on the research data. The alum CPL extract resulted in inhibition zone are showed that the extract owned antimicrobial property. B. Destination. Uh, Good permission will be obtained if the diet contains energy and nutrients that are in balance with your needs. Carbohydrates, protein, and lipids constitute nutrients that must be present in the diet because energy is obtained from this material. Besides, these nutrients must also contain vitamin, mineral, and fiber in order to obtain maximum and healthy growth. In general, the grain grow like wheat, flour. Flour is such energy, and this material can also meet the needs of the use this material in a lot. Tubers to utilize as a source of energy, such as the with cassava. Nuts used as a source of fat and protein, while vegetable and fruit used as a source of vitamin and mineral. Apart from these nutrients to contain it, there are non-nutritional substance or non-nutrition which is usually grouped into the phytochemical group. This, this phytochemical is what has a role maintaining healthy body. Of the roughly 30,000 phytochemicals already known, as many as 5,000 until 10,000 contained in the food. Phytochemicals does include carotene, glycine, and lycopase. Next, I return it into the moderator.
Okay, thank you, Hadi. The next will be read by myself. Okay. C. Formulation of problem. How the antibacterial effect of onion extract against gram is negative. B. Research urgency. A. General purpose. To find out the antibacterial effect of onion extract, bombardment agents extract solely from derived species. B. Special purpose. To find out how to onion extract antibiotic effect Bombay to inhibit to grow of Escherichia bacteria coli obtained from the red species to find out how the onion extract antibiotic effect bombarding with different concentration of inhibition. They grow as secretia coli bacteria obtaining from the pantheons the here to determine the minimum concentration of onion extract bombay whites can produce Escherichia antibacterial effect coli from the pantheons. E. Richard Stargards provides information about the benefits of an extract as an antibacterial extract. The description of an alternative, the extract antibacterial with integrants has the R7 and C obtained, reducing the rate of resistance to antibiotics. Next will be read by Revi Artika.
Chapter 2 Theoretical Review. Good nutrition will be obtained if the diet contains energy and nutrients that are balanced with the needs. Carbohydrates, proteins, and lipids are nutrients that must be present in the diet because energy is obtained from these materials. Besides, this nutrition must also contain vitamins, minerals, and fiber in order to obtain maximum and healthy growth. In general, the group of grains such as wheat flour is a source of energy, and this material can also meet protein needs because of the use of these materials in large quantities. Tubers are also used as a source of energy such as in cassava. Nuts are used as a source of fat and protein, while vegetables and fruits are used as your source of vitamins and minerals. Apart from these nutrients, it also contains non-nutritional or non-nutritional substances which are usually grouped into phytochemicals. These phytochemicals have a role in maintaining a healthy body. Of the approximately now phytochemical as many as 500 to 1000 are found in foodstuff. These phytochemicals include carotene, quercetin, genistein, lycopene. A. Onion Bombay. According to Sutarmi, the treatment of onions in taxonomy is as follows. Divisio Spermatophyta, class Angiosperm, subclass Monocotyledons, family Liliaceae, genus Allium, species Allium cepa L. B. Types of onion Bombay. One sort the varieties red curly, white curly, excel, yellow Bermuda, white Bermuda, Filigrano, and Patna early. Two medium de varieties, Crystal Grano, San Joaquin, California early red. Three long day type varieties, Globe Danvers, Yellow Globe, and Silver King. C. Active components. Onions contain several active components including 1. Amino acid, glutamic acid, arginine, lysine, glycine, and others. 2. Minerals, especially potassium, phosphorus, calcium, manganese, sodium, sulfur, as well as iron, zinc, copper, and selenium in small amounts. 3. Vitamin C, folic acid, and vitamin E. 4. Essential oil, Dipropyl disulfide methyl methathiosulfinate. 5. Quercetin. 6. Allicin with less content than garlic. The benefits. Onions are commonly used to add delicious flavor to certain types of dishes. In addition, this onion has the potential to be used in the medical. I return it to the moderator.
Okay, thank you for the Revi Artika. The next will be read by Cahya.
Chapter 3. Research Method. A. Research Purpose. To find out the inhibitory power of onion extra is effective in inhibiting the growth of Staphylococcus aureus bacteria and to see how much inhibition of onion extra is against the growth of Staphylococcus aureus bacteria. B. Research Methods. Use the control design only task force design group. Where is the onion extra is the extra found by extracting the onion with the maceration method using 96% ethanol solvent. C. Work steps. Wash and chop onions and then aerate it until dry, then in a blender. Then the onion is wedged as much as 100 gr. The first maceration process of onions is mixed with 96% ethanol. Left to stand for 5 days, then filter using filter paper, then measure it again using 96% ethanol solvent for 2 days and filter using filter paper. Then the maceration results were evaporated from the remaining solvent using a vacuum reductory evaporator for 3 hours at 40 degrees Celsius, after which the pure extra was put in an oven for 2 hours and 40 degrees Celsius, then put in a closed sterile bottle and stored in the refrigerator. The tools in this study were sterile in an oven at 170 degrees Celsius and the media were sterile using autoclave at 121 degrees Celsius for 15 minutes. Media brain health infusion brought weight 37 grams and dissolved in 1 liter of distilled water in Erlenmeyer tube, after which the media was sterilized at a temperature of 121 degrees Celsius for 15 minutes. Then poured into a 7 ml test tube. The McParland standard solution consists of 2 components, namely 1% BSL solution and 1% H2SO4 solution. BSL 1% solution of 0.05 ml mixed with 1% H2SO4 solution of 9.95 ml and second until homogeneous. The absorbance value of 0.5 McFarland standard solution is equivalent to bacterial suspension with a concentration of 1.5 multiplied 108. Then the bacteria of time cut into BHIB media and incubate in an incubator at 37 degrees Celsius for 24 hours. Bacteria that were incubated on BHIB media were compact using a McFarland turbidity solution. After that, take a sterile cotton stick and put it into the BHIB media where the Staphylococcus bacteria are squeezed by pressing the cotton stick against the test tube wall, after which it is evenly scratched on the Na media. Cut the filter paper using a perforator with a diameter of 2.5 mm. Soak the dish in each intervention group and treatment group for 30 minutes. After that, take the concreted onion extracts, blend in on part on Na media. Clean machine dishes were placed in another session and 96% ethanol dish as negative control. The same treatment was carried out 5 times on each petri dish. Any laboratory work is carried out near the Bunsen fire to maintain sterility. The 5 petri dishes were incubated in an incubator for 24 hours at 37 degrees Celsius. The diameter of the inhibition zone form indicates the inhibitory power of the onion extract. D. Research result. Based on the results of observation carried out on 5 tests in 5 petri dishes, it, it showed that an inhibition zone was formed around the discs that were given onion extract. The average diameter of the inhibition zone produced by the onion extract. A material is said to have antibacterial activity if the diameter of the resistance form is greater than or equal to 6 mm. Comparison of onion extra and clean machine showed that the diameter of the onion extra was smaller than the diameter of clean machine while the ethanol treated. This did not show an inhibition zone. The results of the diameter of the inhibition zone of onion extra of, of onion extra are smaller than clean machine. This is influenced by the now minimum inhibitory concentration of clean machine, which is 2, while the ability of onions to inhibit bacterial growth is unknown. Clean machine was used as a positive control because it had a good effect on microbes in the mouth. Clean machine can be bacteriostatic or bactericidal. Clean machine as a mechanism to kill the bacteria by preventing protein synthesis from the bacteria. Ethanol 96% as a negative control did not show an inhibition zone. This reinforces the fact that there is no effect of 96% ethanol on the formation of the inhibition zone around the well, which is given onion extra, which in its manufacture used 96% ethanol solvent. Selection of 96% ethanol with the consideration that ethanol is easy to obtain rich neutrally and does not affect the efficacious substance. Well, I ration it to the moderator.
Thank you. Okay, thank you to the Cahya. The next will be read by Yanti.
Next will be ready by myself. Bab 4. Closing. A conclusion. From the result of this study, it can be concluded the dion extract aluminium CBl can inhibit the growth of Staphylococcus aureus bacteria and the conversion of anion extract and Clinda mentioned. So the diameter of the anion extract is less of the diameter of clean clindamation where the ethanol treated this not so abstractly. B. Suggestion. Research hope they picture research can be can out on anion extract at various extract concentration so that the ammonia moment in heavy story concentration leak of the extract can be found against Staphylococcus aureus bacteria. Researchers of that there will be put her research on the inhibition of anion extract as an alternative to dental medicine in the field of the dentistry and the wider community and treating abscession and after our infection. See summary. Bovinians aluminum CPL is an ATP on anion that commonly used in various cuisines in Indonesia. Bulbonians contain antibacterial compounds such as quinones, vaccine allergen. Staphylococcus aureus is one normal microvirus in oral cavity that normally does not cause the disease. The purpose of this study is to determine that the inhibition of Bulbanian extract affected to inhibit the growth of Staphylococcus aureus and to see how much Bulbanian extract inhibit on the ground of Staphylococcus aureus. Things research is an experimental research with keyboard bar dismantle aluminum simple aluminum ball is aluminum sample and been extracted by Merck's method using ethanol 96%. Staphylococcus aureus were taken from the stock of microbiology laboratory Faculty of Medicine Universitas of San Ratulangi, Manado. The result was Actania 85 millimeter with an average of 16 millimeters. From things research can anion extract can inhibit the growth of Staphylococcus aureus. Ammonium CPL anion bark is a cultivation anion extrasynthetically and types of anion. They are often used for various cuisines in Indonesia. Anion monograph with determine the physicochemical characteristic and antioxidant activity. Macroscopic result obtained included asparagos rot and its width and about 9.5 cm long. The stem is for Piosdo and the water is whitish green skindical leaves are dark green. Elongated like an fab and all with and gland of force they manage to in this sick name and the tip is tapered anion tuber are single layer bird but having a diameter of 6 mm, which is equal the anion's macroscopic result contained identification fragment, including hair, covering, and transporting beam with canning, straight and fire. From the result of extract and ethanol extract 70% aluminum CPL. So a drink screeching of 9.669%, 5.16% total consent, 0.07% egg isolably as nedas from 1.10%, water soluble extra and soluble ethanol. Extra 23% vita chemical triogoning contain fun and 3 funds total for flavor contains in ethanol extra of onion tuber was 148 was time 0.12 microgram per gram. Total phenolic contamination absent in anion tuber ethanol extract were an and red 3.47 percent 3. Right go per gram. The antidodradin activity of ethanol extract of anion tuber by the DVVA method recite acidity is 60 from 31 ppm. Reference Dewi take can be see below. Next, I back moderator.
Okay, thank you to Ranti. Okay, this is the result of the percentage of our project assignments. If there are errors, we apologize because we are selfish are also in the learning process. Thank you.
Thank you.